Okay, good morning. I'm going to try to put together a quick video. I'm going to try to keep this in 10 minutes or under, so we'll see how that goes. I want to pick up with what we were lecturing on yesterday, what we were talking about yesterday. I ran out of time. Yesterday we talked about the basic structure of the cell membrane, that it consists of a phospholipid bilayer with polar phosphate heads and nonpolar tails. And whenever water uh, is present, the heads will make contact with the water and the tails will go together because nonpolar stuff is attracted to nonpolar stuff. And polar stuff or charged stuff is attracted to water, and so the water is attracted to these heads. Well, one of the characteristics of this membrane is it is semi-permeable, and that semi-permeability is due to the gaps that exist between the fatty acid legs, and the net result is molecules can pass through that membrane, and they pass through that membrane as a result of osmosis and diffusion, and in this way it is passive transport because it is free. No ATP is required. One of the unique characteristics of the animal cell membrane is the presence of cholesterol, and you can see cholesterol here. I'm circling that in green. Cholesterol is a type of lipid that belongs to a group called steroids, and we see cholesterol in animal cells only. And in some ways, what it does is it compensates for the lack of a cell wall. It maintains the integrity of the membrane. It keeps it fluid, but too much cholesterol can make the membranes rigid and can be dangerous for health, especially in people. Anyway, I want to look at how things move through the membrane, and we talked a little bit about this already. We say that the membrane is selectively permeable, so that basically means that it's porous. So this represents permeability, and that permeability allows some molecules to freely pass through the membrane while restrict the passage of other molecules. So these purple balls, they cannot cross the membrane, and that is because they are just too big. They bump into the holes, but they just can't pass through. In contrast, the triangles are capable of passing through the membrane, and they actually travel in both directions. However, we will find that there is a net movement of triangles going from a high concentration to a low concentration, and we call that net movement diffusion. That does not mean that the triangles do not travel from right to left. It's just they travel that fewer of them travel from right to left than those that travel from left to right. So we get lots of triangle movement going on this side and only a little triangle movement coming back. This movement will continue until some equilibrium point where the concentration of solutes on the left side are relatively equal to the concentration of solutes on the right side. Now, we did a lab in class, and we looked at what happens when we added 3 different eggs to 3 different solutions. We took one egg and we put it into distilled water. We took one egg and we put it into syrup. And we took one egg and we put it into tap water. And I asked you to track the change in mass. And the reason for doing this is so that you could look at how osmosis works. And there is a general rule, and that is that water follows the solute through a semi-permeable membrane. Osmosis is the diffusion of water across a semi-permeable membrane, and water always goes from a high concentration to a low concentration. And what that is a reference to is that we're going from a high water concentration to a low water concentration. So I want to show you an example, and I want to attach some vocabulary to it for you. And here's our example. Let's say we have a solution here that is 10% solute. And if it's 10% solute, that basically means that it's 90% water. At the other side, we have a different solution that's 20% solute, and that means that it's 80% water. So we're going to take this bag, which has a semi-permeable membrane, and we're going to put it into our solution. We're going to wait 24 hours, and we're going to assess, does the bag increase in mass, does the bag decrease in mass, or does the bag stay the same? Well, because the solute concentration is higher inside the bag, we're going to have more water flowing in than we will have water flowing out. The end result is that the bag gains mass, and we would say that the bag is hypertonic. That means that it has more solute, and the solution is hypotonic, and that is it has less solute. As a rule, water goes from a hypotonic solution to a hypertonic solution. So let's try this again. Let's look at another example. Just kind of reverse our numbers. Here, the solute concentration of the cup is 10%, and we have 90% water. And our bag has a solute concentration of 5%, or 95% water. So if we put our bag, try that again, come on. We put our bag into the water solution, the 10% solute solution. What we'll find is that the water will leave the bag because there is more water in the bag than outside the bag. The end result is we can say that the bag is hypotonic, and the solution in which the bag is soaking is hypertonic. And as I told you in the last slide, water always flows from hypo to hyper. So you've got to look at both the solute concentration and the water concentration to make a judgment about the direction the water flows. In the last example, we ask the question, what happens when the two solutions, the solution inside the bag and outside the bag, has the same solute and water concentration? And in this case, we say that it's in equilibrium. This does not mean that the water is not flowing. It just means that the water is flowing at equal rates inside and outside of the bag. As a result, we say that this is an isotonic solution. Isotonic solutions are in equilibrium. Well, with this, you should be able to solve the egg lab and hopefully have an understanding of the direction that water moves when there are solutes present. All right, have a good day. I think that's it. No, why aren't you telling me to save?